So as Posey mentioned, I am Sabrina Heitmann. I am a first-year master's student in Posey Busby's lab, and I am studying the genotype-by-environment interactions of the Populus trichocarpa foliar fungal microbiome. So symbiotic associations between fungi and plants are both evolutionarily ancient and ubiquitous to all plants studied to date. Leaves in particular harbor a hyperdiverse community of fungal microbes that also vary in functionality. Foliar fungal microbes can range from pathogens to mutualists and have been shown to influence host fitness and productivity. Because of these really profound effects they have on plants, foliar fungi therefore likely play a very significant role in plant evolution, community structure, and ecosystem health. Foliar fungi are primarily horizontally transmitted, which means that plant leaves accumulate microbes from the surrounding environmental microbial species pool. However, these microbes are first filtered by assembly processes before it can actually be colonizing the leaf. Abiotic environmental variables such as precipitation rate and temperature can prevent the establishment of different microbial species in a particular environment. Microbes can additionally encounter biotic plant-imposed filters once they actually reach the host. These include plant traits that are controlled by host genes, such as physical barriers or secondary compounds that further limit the realized microbiome. These traits are often plastic and can be influenced by varying environmental conditions. So it's well known that the abiotic environment affects the leaf microbiome, and also plant genetics have been shown to affect the leaf microbiome as well. However, it typically plays a smaller role than the environment. However, less is actually known about how they interact with each other. So in other words, how plant genetic effects change with a given environment. So the motivation for my study is to understand these genotype by environment or G/E interactions on the foliar fungal communities. I use the model system Populus trichocarpa, or the black cottonwood tree, to study these interactions. So Populus trichocarpa is a common and ecologically important riparian tree native to Western North America. It's additionally grown in plantation settings as a desirable biofuel crop due to its fast growth, high cellulose, and low lignin contents. It's the first tree to have its genome sequenced, which has led to a wealth of information that has been associated with it. And this includes a 28 million SNP library of around 1,000 Populus trichocarpa genotypes across their native range. It's also easily propagated, so you can replicate genotypes with cuttings in experiments, and it's easily done. So the ability to use a genetically tractable tree in both natural and plantation settings has made it a model tree for microbial ecology. So a powerful way to explore these plant microbe interactions is to use a reciprocal common garden experiment. By transplanting the same tree genotypes in different garden environments, we can determine if the microbial communities change depending on tree genotype, the garden environment, or their interaction. So in other words, plant genetic material is taken from different natural habitats and planted in foreign and local garden environments. And this results in two different reciprocal common gardens, where one garden contains genetic material from both natural habitats. So what exactly do these genotype, environment, and G/E effects on the microbiome composition actually look like? So in the following slides, I'll depict different hypothetical mechanisms that can cause these effects in reciprocal common garden environments. And these examples are neither mutually exclusive nor exhaustive. I'll be using stomatal length as an example of a plant functional trait that can affect microbial composition in the leaves. So stomata are tiny pores in leaf tissue that allow for gas exchange and can also act as entry points for interior microbial colonization. Stomatal length can act as a microbial filter, so different foliar microbial communities could result depending on the stomatal length expressed by an allele. So let's first take a look at what maybe a genotype effect would look like. So we have two different reciprocal common gardens with the same environmental microbial species pool. Allele A expresses a short stomata length, while allele B expresses a long stomata length. Allele A's shorter stomata filters out the square fungi, while allele B's longer stomata includes it in the foliar fungal microbiome. We see a genotype effect because allele A and allele B have different fungal communities from each other. An environment effect could occur if all alleles express long stomata, but the garden environment contains different microbial species pools. The foliar microbiomes would then be dependent on the environmental species pool, which would indicate an environment effect on the leaf microbiome. G/E interactions can occur if the plant functional trait is plastic or if it has a genetic basis. So in this example, there is genotypic variation for phenotypic plasticity, where stomatal length varies between environments if allele B is present, but not if allele A is present. This results in a G/E interaction on microbiome composition, where allele B responds to the environmental conditions differently. Alternatively, there could be a G/E interaction if there's no phenotypic plasticity for stomatal length, but the environments have different microbial species pools. So we see that allele B, which expresses long stomata, have different microbiomes dependent on the environment. So again, the aim of my research is to use this reciprocal common garden design to study how plant genotype, environmental conditions, as well as their interactions influence the Populus trichocarpa foliar fungal microbiome. So to study this, two poplar reciprocal common gardens were formed in Corvallis, Oregon and Hermiston, Oregon. These gardens are separated by the Cascade Mountain Range, which marks a sharp climatic transition where east of the Cascades is characterized by xeric conditions and west of the Cascades has a wet and mild climate. The different temperatures and annual precipitation in the gardens have also contributed to Populus trichocarpa traits evolving differently east and west of the Cascades. So we want to utilize the genetic differences of these Populus trichocarpa populations. So we used a common garden that contains genotypes from 5 Populus trichocarpa genotypes, or five Populus trichocarpa populations east of the Cascades, and then 5 Populus trichocarpa populations west of the Cascades. And these populations that the trees were collected from represent major populus trichocarpa regional genetic groups that correspond to geographic location and origin. So the genotypes were planted in the Corvallis and Hermiston gardens during the summer of 2018. Each genotype was planted in 12 blocks per garden for a total of 59 genotypes per block. 30 of these genotypes were sourced from the 5 Western populations, and 29 of these genotypes were sourced from the Eastern populations. So the main focus of my research is to characterize the foliar fungal communities found in both of the gardens using DNA metabarcoding approaches. So I collected leaf disc samples this past fall from 1,300 trees in the Corvallis and Hermiston garden. The leaves were cleaned of debris or surface-dwelling microbes, so only the internal microbes would be characterized. I then completed DNA extractions for the samples. And right now, lab work is currently on pause for me, but I am in the process of doing library prep and will hopefully be able to send my samples in for Illumina MySeq metabarcoding in the near future. In addition to characterizing the communities of fungi, I aim to determine whether the potential drivers of these G/E interactions on the leaf microbiome have a phenotypic or genetic basis. So this summer, I plan on taking leaf trait data, which includes measures of leaf mass and area. So if these traits vary based off of the environment, it would indicate that the plants are expressing phenotypic plasticity for that given trait. If the trait varies based on plant genotype, then we know that there's a genetic basis for that trait. So once the foliar fungal sequences are generated, I can analyze how the host, the garden environment, and their interaction affect the leaf microbiome. So first, I think there will be a stronger genotype effect on the microbial communities in the Corvallis garden, and it will be stronger there versus the Hermiston garden. Previous populus trichocarpa research has found less fungal diversity and richness in Eastern environments compared to Western environments. So this could be due to uneven inoculation pressures in Eastern environments due to these dry conditions. If there is uneven inoculation pressure, then plant traits affecting microbiome composition would have less opportunities to act on the microbes. Next, I hypothesize that the environment will have a larger effect than plant genotype in shaping the microbial communities. Studies with populus and other plant species have consistently shown that environmental conditions do play a much larger role in shaping these communities than host genotype. These predicted genotype and environment effects suggest that we will also see a genotype-by-environment interactions contributing to the complexity of foliar fungal microbiome assembly. In the fall of 2019, we decided to actually explore these hypotheses by characterizing the abundance of one of the members of the populus trichocarpa foliar microbiome. Melampsora rust is a common populus trichocarpa fungal pathogen that can cause premature leaf drop and biomass loss, which can then lead to death in natural and plantation settings for this tree. So we collected disease severity data by visually surveying the percent coverage of Uridinea spores, which are the orange-colored pustules that form on the underside of the leaves. And we collected this disease severity on the leaves as a proxy for Melampsora rust abundance. Overall, rust severity was higher in the Hermiston garden than in the Corvallis garden. This is consistent with previous studies that have found that rust is more prevalent east of the Cascades compared to west of the Cascades. Western genotypes had a higher rust severity than Eastern genotypes, so we think that there is a genetic basis for rust susceptibility. And when looking at the genotypes within each garden, we see that the same genotype responds to rust differently in the Corvallis garden versus the Hermiston garden, which indicates that there is a G/E interaction on Melampsora rust. So once my sequencing data has been generated, we'll see if there is a genetic, environment, and G/E effects on the whole fungal communities in the leaves. So foliar fungal microbiomes can play a really large role in plant and ecosystem function in both natural and plantation settings. It's important to understand G/E effects to determine the extent of plant genetic control over fungal communities and how that changes with a given environment. This can inform crop breeding and restoration initiatives about the heritability of the microbiome and if it can evolve as a facet of the host phenotype in response to selection and changing environmental conditions. But the broad category of environment in G/E interactions actually represents the cumulative influence of a potentially huge number of environmental factors. So while my research can determine the degree in which these environments plays a role in shaping the foliar fungal communities, my study isn't designed to untangle which environmental factors are in fact structuring these communities. Instead, my research can be used as a foundation for future manipulative field experiments where these different aspects of environmental complexity can be determined. So with that, I want to thank all of my committee members, the members of the Busby Lab, as well as my cohort for their continued support and feedback throughout my first year. And with that, I'll take questions.
Excellent, Sabrina. Do we have questions for Sabrina?
Yes, I have. How, you just said you clean the leaves. Mm-hmm. How are you cleaning the leaves?
So we used a surfactant, which actually just removes all of the debris on the exterior of the leaves. So it's called tween, and it's a surfactant.
How does that clean the leaf of any microorganisms? There's many that will still adhere.
So from my understanding, it binds to certain molecules and cells that then it releases them once it is washed off of the leaves.
And have you validated this?
I haven't in particular, but there have been studies that have shown that you can, using this method, it is very typical to use for Illumina sequencing. Because instead, so you can also use certain chemicals that, so when you're clearing leaves of surface dwelling microbes, this is the the typical method to use for aluminum metabarcoding.
Jared, I see you have a question.
Yeah, hi Sabrina, nice talk.
Thank you.
I was just curious about your rust part of your talk. So you mentioned looking for G by E. So you saw a difference between East and West, and you related that to a potential G by E interaction. And so I'm just wondering, have you done anything to look at the other part of that three-way interaction in terms of the races of the rust that are present? So if you had different races of rust on the east and west side of the Cascades, then you might get the same effect just because of the fungal, just because of the pathogen, and not necessarily because of a difference in the environment.
Yeah, that's true. There are different races of rust on east and west of the Cascades, so that is a valid point that they might actually be different. So we haven't currently looked at that, but that could be an interesting future study to determine if there are differences in the rust, and then we can relate that back to if we see those similar G, E, and G by E interactions.
Great, thanks.
Other questions for Sabrina?
This is John. I've got a question. Thanks, Sabrina. So you mentioned at the beginning that poplar has this sort of amazing large population of sequenced genotypes, and I guess I'm assuming that the ones in your garden are part of that. Is that correct?
Yeah, so the Western genotypes that were planted in the garden do have SNP data associated with them. However, the Eastern genotypes are still actually getting their SNP data generated, but we might actually be able to get that data in the future.
Okay, right. So I was sort of, thinking about for the future in terms of the possibility of doing some type of GWAS study to follow up on, what comes out of your work. And I'm just curious about sort of thoughts on that and plans.
Yeah, that's definitely been something that we've thought about. There might not be enough genotypes to actually carry out a G by E or a GWAS study, but it would be really interesting to see if there are any genes associated with microbial communities and how that changes if the genotypes are east or west of the Cascades.
Okay, cool. Thank you.
Ian, do you have a question?
Yes. So you were talking about looking at some of the phenotypic variation and like the leaf size and things like that. Is there any way in your analysis to look at like the actual microbial community's influence, if there is any, on instilling some of that phenotypic plasticity, or that kind of just get lumped into the G by E?
We can look at if the leaf traits are associated with the microbial communities, and we can determine if there are these interactions based off of the plant genotype and also where they are planted.
Okay, cool. Thank you.
Any other question for Sabrina? Sabrina, maybe you mentioned this and I missed it, but are you sampling fungi and bacteria in these communities in the leaves, or are you just sampling fungi?
I'm just actually sampling fungi.
Okay, thank you. Any other questions? All right, well, let's unmute and thank Sabrina. Great job.